I'm Dr. Amit Bhattacharya from Ramjas College, Department of Zoology, University of Delhi, and I will be talking about the second part of the lecture, which are the cellular processes and overview. In this part of the lecture, I will be discussing about the various biomolecules of the cell and how exactly they function for the energy synthesis process and how exactly they mobilize the cell for proper functioning and survival. Now, in the cell, there are various biomolecules which are many times referred as the macronutrients of the cell. So, these are the carbohydrates, proteins and the fats, and many of them we consume through and take up through our daily meals and regular meals only. Now, these carbohydrates, proteins and fats, they plays a very important role within our body and in our nutrients parts also. So, the carbohydrates are taken up from various cereals, breads like this one. Then the protein parts, it comes up from the various vegetables, the fish, meat products like this one, and the fats comes from the healthy oils and the various other things. So, it is very important that we have a healthy and balanced eating plate which we should have. So, it should contain say appropriate amount of vegetables over there, the fruit should be there, then the healthy proteins should be present, the whole grains which provide a important part of the carbohydrates which are there, healthy oil should be there, and subsequently the water part is also important. So, if we see the image over here, this particular image shows the healthy eating plate and how exactly these healthy eating plates are important for the necessities and for our body and the biochemical reactions which can take place within the body cells over there. Now, one of the first important biomolecules of the cells are the carbohydrates. So, the carbohydrates are basically many times referred as the saccharides. So, they are the molecular components which are made up of three important elements, which are carbon, hydrogen and the oxygen molecule. Now, the monosaccharides, which includes the glucose, then the disaccharides, which are relatively smaller molecules, often are recognized or called as the sugars, and there are large carbohydrate molecules which are many times referred as the polysaccharide because they are made up of a number of monomeric units of the monomers such as the glucose molecules, and some of the important polysaccharides are starch, cellulose over there. Now, if we see the structure of the polysaccharides which is shown over here, we can see there it contains hundreds of thousands of simple sugars which are there. So, the glycogen, starch, cellulose, these are all polysaccharides which are made up of entirely of the glucose residues. So, each of these glucose residues, they are combined, they are connected to one another with the help of alpha 1,4 linkages or the glycosidic bonds which are shown over there, and if there is any branching, so this branching takes place with the help of 1,6 linkage over there. So, there is a branching which is there. So, many a times these blanched glucose residue long chains can be found in the case of the glycogen molecules over there. Now, in the biomolecule of the cell, in the carbohydrates, if we see over here, the glucose and the fructose, they combine to one another to form a disaccharide which is shown over here, which is called as the sucrose. So, in this dehydration synthesis process, there is a release of the water. So, the glucose and the fructose molecule, they combine. Then there are various other disaccharides, for example, glucose and glucose which forms the maltose, galactose and glucose which forms the lactose over there. So, we can see over here how a simple sugar, it combines to one another to form the disaccharide, then the disaccharide further combines to form the polysaccharides, and how exactly they structure into the complex structures. Now, if we take up the process of the digestion of the carbohydrates, so initially the polysaccharides which we take up through the food sources, the starch, like this one, and the glycogen which is stored within the body when it is broken down. So, they form the disaccharides like the maltose, sucrose, lactose which are there. Then the maltose, sucrose, lactose then further degrades into the monosaccharides. The maltose breaks down into two molecules of glucose, sucrose breaks down into one molecule of glucose and fructose, lactose break down into one molecule of glucose and galactose over there. Then the simple sugar units are further transported into the intestinal cell, and then they are absorbed in the blood, and subsequent processes takes place. So this is how the carbohydrate digestion takes place of the carbohydrate which we consume and which is present within our body. Now, the next important biomolecule which is present within the cells are called as the lipids over here. Now, the lipids are basically classified as the fatty acid, which contains the long chain of carbohydrate chains which terminates with a carboxylic group. Now, over here the palmitate, stearate, which are the saturated fatty acids, are shown over here. So the palmitate contains a carbon which is 16 carbons, so 16 carbons are present over there. The stearate has got 18 carbon which is there. The oleic which is shown over here, it is a unsaturated 18 carbon fatty acid which is there. So unsaturated means there is a presence of a double bond which is there. Now, the various important biomolecules are shown over here which are important to us, like the cholesterol which is there, then a free fatty acid is shown, then the triglyceride is shown, which is a very important part of the fatty acids. The phospholipids are shown over here. So these phospholipids play a very important role in the formation and the structuring of the plasma membrane. So we can see how exactly these lipids biomolecules are also important to us. Then this particular diagram shows the process of lipid digestion which takes place through the various parts of the body, and majority which absorption takes place, it takes place in the small intestines over there. Now, the third important biomolecule of this cell are called as the protein molecules. Now, the proteins are found throughout the body, in the muscles, you can find them in the muscles, bones, skin, hair. Every part of the body or the tissue contains the proteins. Even most of the enzymes are made up of proteins. The hemoglobin, which carries the oxygen within the blood, is also made up of protein. Now, the protein is basically a chain of amino acids. So this is a polyamino acid chain which is there. So each of the amino acid has got a general amino acid structure. So there is a central carbon which is there, hydrogen is there. There is a group R group, the group which is present. Then there is a amino group, and on the other side there is a carboxylic or the acid group which is present. So this is the general amino acid structure of a amino acid which is shown over there. Now, two of the amino acid, they combine with one another to form the peptide bond, and in this formation of the peptide bond, the water molecules is released over there. So we can see over here the one of the amino acid, it is combining with the other amino acid which has got a R2 group. So both of them combine and there is a formation of a peptide bond with the release of the water molecule. So that's how the dipeptide are formed. Then the further combination of the amino acid takes place, and subsequently the chain gets elongated to form a polypeptide chain or polyamino acid chain over there. Now, this particular image, it shows 20 important amino acids which are present there. So some of them are hydrophobic, some are polar molecules over there. So we can see over here some are having very simple, the R group which is there, like the glycine, alanine, valine, like this one. Some of them are having complex structures which are there, like the aspartate, asparagine, like this one. Now, each of the cells, they have got a shared structure. So some of the structures are common in each of the cell. One of the most important part is the plasma membrane. So the plasma membrane, which we just discussed, is a thin coat of lipid that surrounds a cell. So within a cell, there is a thin, around the cell, there is a thin layer of lipid. So these are lipid bilayer which is there. The other part is the cytoplasm, which refers to the cellular material which is present within the plasma membrane, and cytoplasm plays a very important role because it has got many of the enzymes which are functioning for the biochemical processes and the metabolic processes. Then the ribosomes, which are the structures in the cytoplasm which helps in the protein formation, the DNA chromosomes, which are the genetic instruction that the cells needs to make the proteins over there. Now, the lipid bilayer was first given up by the scientists called as Singer and Nicholson. Two of the scientists gave the model system in the year 1972, and many times it is referred as the fluid mosaic model system, which is the general representation of the cell membrane structure and its dynamics. So this lipid bilayer is made up of the lipids which are there. So there is two lipids layers which are there. Within them there are certain proteins which are there. Some of the proteins are present within the lipid bilayer, so they are called as the integral protein, because through this integral protein anything can move from one direction to the other direction. And there are certain proteins which are present on either side of the lipid bilayer, so many a times they are called as the surface proteins over there. So they play a very important role in the dynamics of these particular things. Now the second important organelle within the cells are called as the nucleus. So the discovery of the nucleus and the identification started in the year 1710 over there, but at that time it was not clear that what exactly the nucleus is made up of and what exactly, but it was pointed out that there is a presence of a nucleus. Subsequently, in the year 1829 to 1832, Robert Brown first observed that there is a variety of plant cells and came up with the term which was called as the nucleus. And subsequently another scientist in the 1860s, Frederick Mischer, he discovered the DNA in the pus-soaked badges from the local hospital. So that's how the nucleus name and the identification, the recognition came into the picture. Now the nucleus mostly contains the genes which are present, and it is covered by a membrane which is called as the nuclear envelope. Now this nuclear envelope has got certain pores within them. Now these pores are called as the nucleopores which are there. Now these pores are very important because they regulate the entry and the exit of the molecules from the nucleus over there. Now if we see the image over here, this image shows the structure of the nucleus which is there, and apart from that structure, on the surface of the structure there are presence of nuclear pore complex which is there. So we can see the pore complexes with the help of the TEM image over there, and if we just dissect out this nucleus, we can see that there is presence of nucleus, then there is a nucleolus, chromatin which is present then there is a nuclear envelope, inner membrane, outer membrane which are there, and to this nucleus there is an attachment of the rough endoplasmic reticulum which can be also seen in this particular image. Now this particular chromatin material which is present over there is basically the DNA molecule. Now DNA, as we know that it is a double helix structure which is present. So there are two strands which are moving in anti-parallel directions, and they are joined to one another with the help, connected to one another with the help of the hydrogen bonding. Now the nucleotides which are present within these strands backbone, they combine with the other nucleotides with the help of the hydrogen bonds. So the G bonds with the C and A bonds with the T over there. So there is a complementary base pairing which is happening over there. Now the nucleotides contains the purines and the pyrimidine bases which are linked to the phosphorylated sugars, and the nucleic acid base are linked to a sugar alone is called as the nucleoside over there. Now the nucleotide additionally contains one or more phosphate groups which are there. Now these nucleotides within this particular double helix structure, they pair with one another with the help of a complementary pairing. So there is a formation of a hydrogen bond which takes place between the bases of the opposite strands of the DNA. So there is a specific pairing which is taking place. So the guanine with the cytosine. So we can see with the help of the 3 hydrogen bond, the guanine and the cytosine are paired. Then there is an adenine with the thymine, in which the adenine and the thymine are paired to one another with the help of 2 hydrogen bonds over there. Now apart from that, in the cell, within the cell there are various important functioning which is taking place. So there is a DNA which contains the various genes. Genes means the exon, introns which are present over there. Now the first thing which happens within them is the transcription. So the DNA strands, they get unfolded and the transcription takes place. Now the RNA is formed. Now subsequently the RNA is spliced so that the introns are removed and all the exons are attached to one another to form out the mRNA, and now this mRNA, it attaches itself to the ribosomes, which are the specialized molecules present within the cell which are responsible for the synthesis of the proteins, and and depending upon the coding structures which are present within this mRNA, the amino acid comes and combines to one another to form the polypeptide chain, which we called as the amino chain over there. So once the amino chain is formed over there, then there is a post-translation modification which happens. What it does is it causes the proper folding of that particular protein, so that this protein now becomes functional over there. Now this protein can form into any kind of enzyme over there, any signaling molecules, or the various other functioning it can get into. Now this particular table is showing the DNA contents of the cells. So in the bacteria, various prokaryotes, then comes the unicellular eukaryotes, the plants, animals. So we can see over here the haploid DNA content as well as the number of genes which is present within these particular organism is shown over there. Now the nucleus, as we just discussed, is a specialized membrane enclosed organelle within the cell that contains the chromosome. So we can see over here how the DNA is transcribed into mRNA, then the mRNA moves out. It is then translated with the help of the ribosomes, the proteins are formed, and then the proteins are sorted within the endoplasmic reticulum to form what kind of protein has been formed, and depending upon that, it is then compartmentalized or localized in that particular part of the cell. Now the next important organelles within the cell is the mitochondria. So mitochondria is basically the membrane bound cell organelles, and which generates the most of the chemical energy needed to power the cell's biochemical reaction. So we can see over here the mitochondria is made up of two membranes. There is a outer membrane which is there, and there is a inner membrane which is there, and apart from that there is presence of mitochondrial matrix which is present within them. Now if we see, the mitochondria is basically the center of cellular energy metabolism which is taking place. So in this particular image, if we see a cell has been shown, in the cell, in the cytoplasm, the very basic reaction like the glycolysis is taking place, where the glucose is broken down into the pyruvate molecule, and then the pyruvate is entering into the tricarboxylic acid cycle which is taking place within the mitochondrial matrix. And in the tricarboxylic acid cycle, the electrons are being released. These electrons are taken up by the electron accepting carrier molecules which are present within the electron transport chain, or many times referred as the oxidative phosphorylation reaction, and by sequence of reaction which involves the various complexes like the complex 1, complex 3, complex 4, like this one, the electrons moves from one complex to the other complex, and finally these electrons are taken up by half a molecule of oxygen to form the water molecule, and thus in this particular way the energy is being released which develops the formation of the ATP molecule with the help of the ATP synthase or FOF1 ATPase enzyme molecule which is present or connected with the electron transport chain. So that's how the ATP is formed over there. So we can see over here there is a very synchronized reactions which are taking place and which are taking place with the one another and present and doing at the various parts of the location of within the cell. Now within this cell there is another important organelles which is present, which is called as the endoplasmic reticulum. Now the endoplasmic reticulum is basically a network of the membranes which are present within the cells, which is mainly involved in the assembly and the sorting of these particular proteins which is taking place. The endoplasmic reticulum have been classified into two major forms. One is called as the smooth, the other one is called as the rough endoplasmic reticulum, depending upon the production of the protein. So if there is an attachment of the ribosomes to the endoplasmic reticulum, they are called as the rough because they gives a representation of roughness on the smooth surface of the endoplasmic reticulum. If they are absent, then they are called as the smooth endoplasmic reticulum over there. Now the ribosomes are also referred as the protein factories which is present within the cell. So these ribosomes are made up of ribosomal RNA and the proteins, and they carry out the various protein synthesis majorly in two major locations. One, the cytosols where the free ribosomes are present, and on the outside of the endoplasmic reticulum where they are attached and they are called as the bound ribosomes. So there are free ribosomes which are also there, and there is a bound ribosomes which are also there. Now these ribosomes contains two units. One is called as the large subunit, the other one is called as a smaller subunit. So if we see the transmission electron microscopy image over there, so we can see over here the ribosomes which are attached onto the endoplasmic reticulum, which are shown. Now each of the ribosomes contains a large subunit, as well as there is a small subunit which is present over there. Now the endoplasmic reticulum are basically referred as the site for the protein synthesis, because there is a presence of ribosomes within them. So if you see over here, the nucleus is shown over there, where the nuclear pores can be seen over there. So apart from that, there is the attachment of the endoplasmic reticulum on which the ribosomes are attached. So this is called as the rough endoplasmic reticulum. Now the ER and the Golgis are involved in the modification and the sorting of the proteins which are being synthesized on the rough endoplasmic reticulum. So in this particular image, if we see, the proteins are being formed on the rough endoplasmic reticulum, and then they are sorted at the various location within the endoplasmic reticulum, and they are released over there. Once they are released, with the help of the vesicles, they move into the various phases of the Golgi complex, which are called as the cis and the trans phase of the Golgi complexes, where they combine and they releases within the Golgi complex. Then the sorting takes place, and we know that the proteins can be enzymes also, proteins can be the signaling molecules also, they can form the receptors which are present on the surface of the cell also. So depending upon what kind of protein it is, so the sorting takes place. If it is a surface receptor molecule, with the help of the secretory vesicle, it moves to the outer layer of the cell and they form the receptors. If it is a enzyme, like for example the lysosome enzymes, they are engulfed and blebbed with the help of the secretory vesicle, where they form the lysozyme over there, or the digestive enzymes over there. So the endoplasmic reticulum and the Golgi, if we can see, also functions in a very harmonious way, so that the proteins are being modified and sorted in a proper way. Now the cells, they divide by a process which is called as the cell cycle or the division phases. Now the cell cycle is divided into two phases. One is called as the mitotic phase, the other one is called as the interphase. Now in the interphase, basically the cell prepares itself for the division process, and while in the mitotic phase, the division actually takes place. The interphase have been classified into G1, S and the G2 phase over there. So in the G1 phase, the cell prepares itself for the synthesis. So the DNA single structure, it synthesizes into double helix structure. So the synthesis happens over there, and in the G2 phase, now these particular chromatin material, they condense itself and prepare itself for the mitotic phase or the M phase over there. Now the mitotic phase is further divided into various phases over there. So if we see over here, this is the interphase which is shown, where the cell prepare itself for the division process, and basically if we focus into the nucleus over there, the nuclear or the chromatin material is dispersed in the case of the interphases, while in the prophase, if we see over here, they have been condensed and sorted into chromosomal structures, and then comes the mitotic phase, where the division takes place. So in the mitotic phase, there is a metaphase which is there, then there is a anaphase and the telophase which is there in these particular phases. So in the metaphase, if we see, all these chromosomes, they get arranged in the equatorial plane, and they get connected with the help of the microtubule, which is called as the spindle fiber. So these spindle fibers get connected at the center of the chromosomes, which is called as the centromeres. So these spindle fibers get attached over there, and that's how the metaphase is there. So everything we can see, all the chromosomes are arranged in the equatorial plane. So they are present in the equatorial region. That's why it is called as the equatorial view or equatorial region plane view also. Now once the metaphase has occurred and the spindle fibers have got attached to the center of the cell, which is the centromere, then the shortening of the spindle fibers start taking place. So as the shortening of the taking place, the chromatid arm of the chromosome, they get separated to one another and they start moving into the opposite poles. So now they have started moving into the opposite pole. So this is the initial phase of the movement. So it is referred as the anaphase, where they are moving towards the opposite pole. Now if we see over here in the next phase, there is a cleavage furrow which is formed over there, and subsequently from one cell, two daughter cells are formed, and each of them contains a copy of that particular chromosome which is formed over there. So we can see these cells divides with the help of the specialized functioning which is called as the cell cycle, and this cell cycle is divided into two phases. One is called as the interphase, the other one is called as the M phase or the mitotic phase. And in the interphase, the cell prepare itself for the division process, while in the mitotic phase it divides over there, and finally when the division takes place, the two daughter cells are formed. Now once the daughter cells are formed, they start functioning and developing in a normal way, and once the time comes for the division of these daughter cell, again they move into the other phase, which is the they get into the cell division phase of their particular action. So that's how these particular reactions takes place within the cell. Now if you see the image over here, this is an image which shows the metaphase, in which we can see all the chromosomes are arranged in the equatorial plane over there, and we can see the other cells which may be in the initial phase or the prophase or the interphase of their particular reaction. Then we can see over here, this is an image which shows the anaphase over there. So we can see the chromatid arm of the chromosome, they have got separated and now they are moving towards the opposite pole. So that causes the anaphase over there. So this is what is happening in this particular cell cycle. Now with this note, I come to the end of this lecture. So in this part of the lecture, we discussed about the various biomolecules of the cell, how exactly they are important and what are the biochemical reactions which takes place, what are the cell organelles, important organelles which are there, and then we discussed about the division of the cell, how they divide with the help of the cell division phases which are divided into the interphase and the mitotic phase. So see you in the next lecture. Thank you very much.